So I'm going to present my project, which is about the analysis of hybridism and introgression using next generation sequencing. It is supervised by Professor Joachim Hermisson at the University of Vienna. So an hybridism is defined following Barton and Hewitt as a narrow region in which genetically distinct populations meet, mate, and produce hybrids. And there is quite a few examples of hybrids in nature, for example, between these two hairs, or between these two flowers. Janine will speak a bit more about this one, or between these two worlds, for example. In this definition, they use genetically distinct population and not species. Because if you refer to the most commonly used definition, it is a biological species concept of mare, which defines species as a group of individuals that can interbreed. And if you follow strictly this definition, any populations that produce hybrids and hybrids are just one species and not two. But the problem is, if you look at a trio of speciation, like this one, for example, at the top it's obvious you have one species. At the bottom, it's obvious you have two species. But what happened here, it's not very clear. And this definition does not help at all for this, defining what happened here. And hybridism can be an explanation for this phenomenon, because if you have an hybrid reduced thinness, then it may explain why you have this. If migrants first move in these hybrid zones that have the reduced thinness, they won't be able to produce lots of offsprings, and these offsprings will have a really bad thinness, so they won't be able to move in the other population. And then you will have this divergence between these two populations. And that's this one kind of question we want to investigate. So I will just present my first project, which is about the accumulation of genetic barrier in face of gene flow. So in the absence of gene flow, it is known that you can accumulate, because of local adaptation and drift, lots of incompatibilities between the two genomes, and then if you put them back together, they can't mate anymore. But when there is gene flow, it's not exactly known. So this picture is from Michel et al., and this represents the genetic divergence along the genome. And so you can have some high level of genetic divergence, which is mainly due often to a gene under divergent selection or local adaptation. And otherwise it's just low level. And the question is, when you have this in the genome, how can you just increase the genetic divergence level to obtain two different species? And there has been lots of verbal theory to explain this, but not true analytical model. And one explanation is you just accumulate mutation here, and you just extend this like this. Another possibility is just you have a new mutation that happens somewhere else in the genome, and that may interact with this one. And so, because of this epistatic interaction, you can have a new island that appears somewhere else in the genome. Once you have enough islands, you just have the level of divergence of all the genomes that is high enough to have these two species. And to explain this, to represent these incompatibilities, we are using the Belson-Dobzhansky-Muller model. The idea of this model is that you are considering only two loci, and you have two new mutations. At the first loci, you have the blue one. At the second loci, you have the green one. Both are advantageous, as you can see here. Here is the thinness, and here is the level of the white type. And then if you put them together just by sex, you expect to produce a good, very good individual, because you will put the two effects of the mutation together. But it's not the case. You have something really bad. And that's the idea of these incompatibilities. This is this epistatic effect that you put two mutations that are good together and you have something really bad. And this is a bit more formal way to present things. So the uppercase letter represents the new mutation. So this is the first one, this is the second one here. So both of them are good. And then this is what happens when you put them together. You have this epistatic term here that appears, that produces strongly this thickness. And we are using a continent island model to represent these things. And this is what we are studying. So now you just look first at the continent here. You have two loci, and one is a continental type adaptation, the big B. It appears on the continent, it is adapt on the continent. On the island you have the big A here, which is an island type adaptation, so it's good on the island. The big B comes from migration from the continent to the island. It can be good or bad. And then you have these two. When you put them together just by sex, you have these hybrids that are really bad. And this case has already been studied by Claudia Bank and Joachim Hermisson. And they have shown that in that case you can create a genetic barrier. Of course, it won't be strong enough to stop gene flow along the whole genome, but at least it will reduce this locally. And the question we want to address now is, how can we improve this genetic barrier? And one way to study this question is just What happens if a new mutation appears? So it's an unusual site, so now you consider a pseudo-site model with this small c here. And the new mutation here, it represents one case, but it can also appear here, or here, or here. And what kind of properties this new mutation should have to increase this genetic barrier and to reduce the effective gene flow between the two populations? We can also consider case of mutation on the continent, of course. And in that case, if it will be a continental type adaptation, which means that it will interact with big A, whereas the big C just on the side before here appears on the island and is an island type, so will interact with the big B. So in both cases, we can show that for a given range of parameters, we can increase the genetic barrier. So this idea you can still grow this genetic barrier, increases of gene flow is possible. Now I will just introduce quickly the next two projects I'm going to do. So the second one will be the analysis of an hybrid zone between two global species with janine. One of the questions we want to address is to determine whether this has been formed in a parapatric speciation scenario or in an allopatric scenario. So in the parapatric speciation scenario, it means that you have first the two populations that live close together and that can even overlap. And then because of this accumulation of incompatibilities I have just shown before, you have these hybrids that have reduced thinness, and they will act as a well in a way to prevent migration between the two populations and just let some divergence accumulate in the two genomes. The secondary contact scenario is just that you have the two populations that are truly separate, there is no migration, they just evolve on their own, and then you put them back together. They are divergent enough to not be able to mix anymore, but not enough because they are still able to mate and to produce this hybrid. And my last project is about the accumulation of mutation. What is this effect? Because there is always the risk of losing this island adaptation. Because you have in this model, you have this island that is small and the continent that is big. So it means that you will have a lot of mutation on the continent and a few on the island because of the population size. So you will accumulate lots of continental type mutation, so lots of incompatibilities with a few island adaptation. And this will cause a risk of losing the island adaptation because it will have to face too many incompatibilities. And one solution, and one of the questions we want to address is how often should we have a new mutation on the island to rescue the previous one in a way and to prevent the swamping of the island adaptation by the continental one. And this asks the question of what should be the relative size between the two populations. Thanks for your attention.